Hello and welcome to Psyched, the show where we explore psychedelics through social, economic, and political perspectives. Our next speaker is Chris Witowski. He will be talking on a new era in psychedelics, pharmacology, and the future of mindful medicine. Christopher G. Witowski. Chris has always been fascinated with nature and the benefits it supplies to humanity. His first research project was assessing lipid profiles of microalgae on an algae-to-biofuels grant. From there, the main focus of his PhD studies was to cultivate fungi and bacteria using innovative techniques to increase their bioactivity and therapeutic potential. He transitioned into the burgeoning cannabis industry as the chief scientist and leader for multi-state operated AltMed. Under his direction, 50+ distinct cannabis-infused products have been developed and launched, receiving critical acclaim. The Mu brand features pharmaceutically developed patches, gels, inhalers, and oral delivery systems. His career research has led to a patent portfolio of anti-infective natural products, medicinal chemistry, as well as cannabinoid compositions and methods of use. Psychedelic molecules such as psilocybin further characterize the reemergence of natural products as holistic medicines. The core mechanism of these neuromodulators will revolutionize our understanding of mental health and treatment of these afflictions. He co-founded Psilera Bioscience with the aim to develop new drug-based therapies to tackle mood disorders, cognitive diseases, and addiction. In its early stages, Psilera has secured seed capital, engaged with research collaborators, and established intellectual property for new formulations and therapies. Chris, thank you so much for joining us, and welcome to Psyched.
Thank you, Merritt, for the introduction, as well as the invitation to the conference. I will say so far I've really enjoyed the speakers and the engagement, as well as the content, and we're very excited to be here. Before I begin, I'd like to introduce myself. Again, I'm the co-founder and CEO of Psilera Bioscience, and like many others, I've been personally affected by mental illness, having watched my grandmother go through a 10-year battle with Alzheimer's disease, having other close family members have bipolar disorder, depression, and traumatic events that have led to crippling anxiety. And I've always been fascinated by the brain and neurology, and I think we're really just only scratching the surface of some of these new developments that have occurred. And psychedelics not only are a method to not mask the symptoms, but actually target the underlying condition to cure these afflictions with only 1 or 2 doses. And being a researcher and a scientist, really this is a very exciting time to be involved in this type of research, and one that I'm personally very attached to and very passionate about. So with that, I will attempt to share my screen. Hopefully everyone can see it.
Yep, you're good.
All right. So today I'll start by talking about natural products and their medicinal aspects, go into some of the different psychedelics that are in the clinic, and talk about some of the limitations and obstacles from psychedelic research. And then I'll introduce Psilera Bioscience, go into our research strategy and methodologies in order to create new treatments for neurological disorders. So natural products have been around much longer than we have, initially starting out as sort of a food supply for ancient cultures, and then kind of with traditional medicine, incrementally getting into more of the therapeutic aspects of natural products. And we'll take cannabis, for instance. Its use goes back at least 10,000 years, and mushrooms as well in traditional medicine. With indigenous tribes all over the world. Really, this long-term use correlates to a really good safety profile, which from a drug discovery and development standpoint is crucial to have that safety and efficacy. And now we're really starting to see a reemergence in natural products. And for a long time, the pharmaceutical industry was more focused upon synthetic compounds to treat diseases. And for now, we're starting to see that shift back towards natural products. And I think cannabis and psychedelics as well are kind of reinforcing this notion that we've had all along, that natural sources really are a good starting point for drug discovery. So as a PhD researcher in natural product drug discovery, I was able to meet the professors Newman and Cragg, and they're kind of the godfathers of natural products research and natural products drug discovery. Every 4 years or so, they put out a review article looking at all the new FDA-approved entities and determining whether they're a biologic, a natural product, a derivative of a natural product, or purely synthetic. So if we go back from 1981 until now, about 65% of all FDA-approved small molecules are derived from natural products. Again, they have their limitations in terms of reproducibility and supply, which is why they do serve as a starting point for kind of future synthetic studies in order to optimize their properties. So looking at some of the different targets that psychedelics affect, serotonin, 5-HT is a common abbreviation that I'll mention a couple of times today. This is really the major target that a lot of the classic psychedelics like the tryptamine, psilocybin, DMT, LSD, mescaline, and obviously the serotonin system has long been targeted for depression, but as well headaches, nausea, and as well as cognition. The NMDA system, this is largely responsible for ketamine and its effects. While it is a dissociative, it is also used for other neurological disorders such as Alzheimer's disease and Parkinson's. And then dopamine can be affected sort of downstream from some of these psychedelic targets, such as LSD and MDMA. So I told myself I wouldn't get too hardcore into chemistry. When some people see these chemical structures, it does tend to make their eyes glaze over, but I've done my best to kind of highlight the major portions that I'm talking about. So in the top left, you have serotonin, and this is the endogenous molecule that your brain produces in order to make you feel good. And again, 5-HT is sort of the abbreviation for serotonin and the serotonin system. And there are 14 serotonin subtypes, 5-HT1 through 7, ABC, etc. And all of these do have beneficial aspects for a wide variety of treatments. So just looking at the triptan drug class, these are 5-HT1B agonists, and these have been approved and researched since the 1980s for headaches and migraines. So if you look at the red portion of the molecules that I've highlighted, apologize, you see similarities between serotonin and the triptan drug class. The major thing that I'll point to is sort of the 5 substitution that you see off the triptan drug class, which mimics serotonin. However, when we go to psychedelics and the 5-HT2A agonists, the substitution on the phenyl ring of the indole ring really determines more selectivity towards the 5-HT2A receptor type. DMT having no substitution, psilocybin, psilocin, and LSD all have substitution at the 4 position. So minor changes really can make a big difference in the selectivity and the targets of these drugs. So now moving into the compounds that are in the clinic or have progressed through it, we'll start with ketamine. It is a synthetic molecule that has been around for a long time, both for anesthesia as well as other analgesic properties. And recently in 2019, S-ketamine, which is the S enantiomer of ketamine, was approved for treatment-resistant depression. And the treatment type for ketamine really kind of paved the way for the current psychedelic research, in which you have sessions both before and after, in between drug sessions, where you're supervised by psychiatrists and other mental health professionals. The treatments have to be administered in the clinic. And I would say the antidepressive capabilities of ketamine aren't by themselves maybe a blockbuster, but really with most legacy antidepressants, you have a 2 to 3 month kind of lag time between start of treatment to efficacious benefits. And what ketamine does is it really cuts that down to about 24 to 48 hours before starting to have a good benefit. So really on this data alone, kind of ketamine really transformed the antidepressant market and allowed for new treatments for neurological disorders. Ketamine, since it does have to be administered clinically, does associate a higher cost with the sessions and having recurring doses. And I'll touch on that a little bit later on. So moving now to MDMA, another synthetic compound being championed by MAPS, the nonprofit. It is currently in phase 3 for PTSD, again, clinically administered. And the drug experience itself, along with a lot of the other psychedelics, isn't the only part of the therapeutic benefit. You have to have psychological therapy in order to really get to the bottom of the root cause of the problem. And having the psychiatric treatment really helps do this. And with MDMA, they showed about 80% of their patients did not meet the criteria for PTSD after 1 year. So really, really great work by them being done at MAPS. And lastly, psilocybin. It is a prodrug. So psilocybin is the natural compound produced by mushrooms, a wide variety of mushrooms. And you do see the phosphate group on the 4 position of the molecule. And when you ingest this compound orally, the acidity and the enzymes in your stomach actually dephosphorylate this into psilocin. And this is the active ingredient which has the neurological benefits. So also when you look at the magic mushroom itself, you do get a blue bruising phenomenon. And this is actually due from the conversion of psilocybin to psilocin. And this can be done enzymatically or oxidatively. And the blue color is associated with psilocin as well as the quinolone intermediate that is produced through this pathway. Currently it is in phase 2B clinical trials for treatment-resistant depression. Again, a similar kind of clinical approach where you have to be inpatient for the therapy. But what initially we've seen so far with the data from Compass Pathways is the efficacy is much greater than what we see with ketamine. It has a longer lasting effect and still has sort of the rapid onset of efficacious benefit. So I talked a little bit about some of the costs that are associated with these treatments. You do have to have before and after sessions with psychiatrists. You generally have at least 2 mentors there and guides who go through the experience itself. And then you do have kind of recurring doses and therapy sessions in order to retain the therapeutic benefit. And all of these kind of combine into an increased cost. And what we're seeing currently is a lot of out-of-pocket costs. I do think in the future you will see insurance companies start to take on more of these, but these are relatively new and innovative therapies, and they do kind of pass through to the individual patient and healthcare providers. Another big limitation that I'll mention are the exclusion criteria for a lot of these psychedelic therapies as well as clinical trials. And a big one, since psychedelics do increase your heart rate and blood pressure, are these coexisting conditions. So those with high blood pressure aren't able to participate due to some exacerbation of these comorbidities, as well as a past history of psychosis or schizophrenia. And psychedelics can really enhance and potentially exacerbate some of these pre-existing conditions, which is why they're excluded. But I think really what we're trying to do, and as a community trying to do, is to increase psychedelic knowledge across many people and trying to increase access, whether it be pharmaceutical or otherwise. And these pre-existing conditions really kind of, we're looking at a small subset. And to further upon some of these demographics, which we have talked about a little bit during the course of this conference, is the demographics of the trial participants. A meta-analysis in 2018 looked at 18 of the psychedelic clinical trials, 282 participants, and 82% of these participants were white. Again, looking at the meta-analysis of 6 psilocybin trials, you had 143 participants, and over 90% of these people had a college degree or greater. Looking at some of the racial disparities that we've seen, not only over the last couple of weeks, but, for a long time, we really need to expand upon kind of our current vision of, the clinical outcomes. There can be physiological differences from different genders and different races. And in an era where really personalized medicine is becoming more prevalent, and really to expand upon this, we really need to look past largely white educated participants. And, looking at some of the demographics we see now, it is a little troubling, but I do think over the long term, you will see this be beneficial for many and all people. So we looked at some of the obstacles involved with the current trials, and I would say one of the big ones is the stigma that is still involved with psychedelic and psychedelic research. And I think we can draw some experience from the cannabis industry, and we really touch on the efficacy and the beneficial outcomes and not so much the recreational use of these compounds. I think that's really going to go and improve the public outlook upon these. Another way that we could potentially increase acceptance of psychedelic therapies is by trying to reduce the psychoactive activity. Microdosing obviously is one of the main ways that you can do this. By reducing the dose, you reduce the psychological effects. But as well, I mentioned there are about 14 different serotonin receptor subtypes. So if we're not just looking at 5-HT2A, which gives you sort of the psychoactive effects, we could open up therapies to a lot more conditions and a greater acceptance in patients who necessarily can't have the psychedelic experience themselves. And another point I think we should really touch on is the duration of effects. So going back into the cost, generally the inpatient therapies are 4 to 8 hours of clinical supervision. But if we can reduce this to maybe a couple of hours, we can really cut down on costs all the way around. And DMT is a very good example of this. So ayahuasca contains DMT as well as monoamine oxidase inhibitors, which allow DMT to be consumed orally and absorbed without being otherwise destroyed. And when orally consumed, you have a duration of effect for about 4 to 6 hours. But DMT can also be vaporized or smoked. And the experience of this only lasts about 15 or 20 minutes. So especially the longer the treatment, the more potential you have for bad trips or the challenging experiences. And by greatly reducing the time of the psychedelic experience, we could potentially reduce the side effects and having bad outcomes, which for certain populations, maybe those challenging experiences really aren't beneficial, especially for those with other psychological disorders. And again, I think by using these techniques, we can open up psychedelic therapies to more and more people. And that is our goal at Psilera Bioscience. And this is a biotechnology company co-founded between myself and Dr. Jackie Salm. Together, we have over 20 years of natural product drug discovery experience. From our academic time, we have over 17 peer-reviewed publications and 7 patents. Many of these came from our 8 or so years in the cannabis industry. And our major focus on those patents was developing new bioavailable delivery methods using cannabinoids and using this technology to put into drug delivery devices such as oral delivery systems, nebulizers, non-combustible meter-dose inhalers, as well as transdermal gels and extended release patches. We also have a board consisting of pharmaceutical executives and powerful partners with our collaboration at the University of South Florida. So looking at our pipeline, We have submitted provisional patents on new chemical entities, and we will be using these as a target validation with some of our researchers at USF and the conditions listed. And we're going to leverage our experience with drug delivery technologies. And if you look at the bottom of the screen, we see a recurring dose method where you have kind of the peaks and valleys of a recurring dose. And with the peaks, you can have some of these psychedelic effects, but as it tails off into the valley, that's when you start to be below the therapeutic threshold. But by creating controlled delivery systems, we're able to maintain a very consistent dose over time, above the therapeutic threshold, but below some of the psychoactive benefits. And again, I think by using these technologies, we can really open up the course of treatment. So quickly looking at some of the anti-addiction potential, and most of this research has come from Johns Hopkins and the researchers there. So nicotine and smoking with the 20 to 30 milligram psilocybin session, they showed that 80% of participants had quit smoking at 6 months. And at 1 year, 60% of those had quit smoking. And I believe the current cessation rates of smoking with what's currently on the market is about 35% or so. So, there is a lot of promise here. And you see similar outcomes with alcohol, where heavy drinking days and drinking days were down 7 months after psilocybin treatment. And, these compounds really disrupt the default mode network and get into these repetitive patterns of either substance abuse or, negative thoughts that are associated with depression. And there's a wide variety of effects that can be attributed to a lot of these psychedelics, and one that definitely requires further research. And lastly, cognition, as I mentioned, I have some personal experience with these. And microdosing has become a pretty common use, especially Silicon Valley, and kind of now really has been quite prevalent. And this is supposed to increase productivity and creativity. And it's well known now that 5-HT2A pathway is very important for the learning and memory creation, which ties in very well to disorders like dementia and Alzheimer's disease. The tau and beta amyloid plaques, these are kind of the current target that a lot of pharmaceutical companies have been looking at to cure Alzheimer's disease. There's now evidence that shows these psychedelics are able to modulate some of these plaques, as well as target neuroinflammation. These compounds are very, very potent anti-inflammatory agents, especially for the brain, for things like traumatic brain injury, as well as inflammation is one of the early onset indicators of Alzheimer's disease. And especially neurogenesis, the ability to recreate and connect new neural pathways that would otherwise go unlooked. And again, psychedelics are a really burgeoning source of new targets here. So in summation, I hope I've done a decent job of trying to explain the importance of natural products to medicine, and a starting point for drug development. And I think this ties in very well to the industry that we're seeing unfold before our eyes, and whether these compounds are developed by a synthetic route or more on their natural product side, I do think you're going to see a lot of efficacious outcomes in the coming years. Talked about some of their progress in the clinic to date. There are some limitations to this research, but overall, we're seeing a paradigm shift in neurological research due to psychedelics, and I don't foresee that slowing down anytime soon, let's say. And lastly, I talked about Psilera Bioscience and our approach to creating new neurological treatments. And if you have any questions, feel free to visit our website at psilera.com or email me directly at chris@psilera.com. And I'd like to thank you all for taking the time to listen to me today. And I look forward to any questions. So thank you very much.
Hey, Chris, thank you so much for sharing. Really appreciate it. Our first question comes from the audience. It's given your experience in microbial strategies for biofuels, can you comment on the synthetic biology strategies being applied to the psychedelic field?
It's a good question. So some of the psychedelics will say, psilocybin, going from a fungal species to a yeast species is actually a pretty easy conversion. For things like, we'll say cannabis, going from a plant to a yeast, it's a little more challenging. I do think there's beneficial aspects that can be done through this translational research. But again, the starting material itself is a good biological rich resource. And actually some of the research I did as a graduate student was looking at different cultivation techniques in order to upregulate or completely change the metabolic profile of various fungi, some of which were culturing more than one microbe per flask, so that you can, create new chemistry that would otherwise be unlooked in a single organism, in a single, vessel. And we're able to produce new chemistry using these types of techniques, and especially selective. We were doing a lot of antimicrobial type research for these. So, again, I think there's a lot of avenues that are worth exploring in this field.
Mm-hmm. Over the next 5 years, 10 years, where do you think the majority of the most impactful research will be coming from? Do you think it's going to be the for-profits that are forming today, a continuation of the nonprofits, partnerships and collaborations that come out of the two? We'd just love to hear your thoughts.
I really hope that it can be a collective benefit across all of those, because, being an academic researcher, you can almost call those nonprofits, and they really serve as sort of a spark or a starting point for a lot of this development process, which are picked up by for-profit industries and pharmaceutical companies. And again, I have to give a lot of credit to MAPS in doing what they're doing as a nonprofit. It's taken them some time, but anything is possible, and we're seeing that now.
Mm-hmm. One of our other audience questions is, could you just give us a little bit of a perspective on Psilera as opposed to other psychedelic startups that are maybe, tackling some of the same challenges that y'all are looking at?
Yeah, you can hear a lot of the same pitches per se. But, I feel really confident in our team. Myself and Dr. Salm are, active, engaged researchers. We're not trying to outsource any of our development. We're doing it in-house. We bring a lot of experience, from the drug discovery standpoint, whether that be, purely natural product fungal research, or more of a, potentially scientific, synthetic drug development. And, a lot of our focus is going to be on different ways to deliver these compounds to open up more options for more patients.
Mm-hmm. Another question that we got from the audience is, do you know of, or are you planning on looking at sex differences within 5-HT receptor impacts on efficacy of psychedelics for depression and PTSD, et cetera?
Yeah, personally, we're not quite at that stage yet where we can do this research. I do think, I think there is a gender discrepancy between kind of the receptor density of serotonin across obviously gender and other things. Not one that we're looking at currently per se, but, one that I do think needs some in-depth research on.
Mm-hmm, definitely. Well, thank you so much for your time. We really appreciate it, Chris, and looking forward to talking more offline.
Thank you, and thanks everyone. Bye-bye.